All living organisms are made of cells and their products. The cells are the structural and functional units of life. In this session, I'll be dealing with cell cycle. The cell cycle is a sequence of events during which the cells prepare to divide to give rise to two daughter cells. A cell cycle consists of 5 stages. They are G1, S, G2, M and D phases. The G1, S and G2 phase constitute the interphase. Thus, there are 3 main phases in the cell cycle. They are the interphase comprising of G1, S and G2, the mitotic phase or the M phase, which includes the karyokinesis, and cytokinesis or the D phase or the cell division phase. The first stage in cell cycle is interphase. It is the longest phase in the cell cycle. It comprises of G1, S and G2 phase. G1 or gap 1 phase is the first stage in interphase. During this phase, the cell prepares for synthesis of tRNA, mRNA and proteins required for DNA replication. In the S phase, the next phase is the S phase or synthesis phase, where there is DNA synthesis. During this phase, replication of DNA takes place. Since histones are also synthesized, there is replication of chromosomes too in this phase. Following the S phase is the G2 phase. During this phase, the cell grows as well as the organelles also grows. The cell now prepares for the next phase, that is mitotic phase. The mitotic phase. In the mitotic phase, the division of chromosomes takes place. It is divided into 4 stages. They are the Prophase, the Metaphase, Anaphase and the Telophase. The mitotic division gives rise to 2 daughter cells which are identical to each other and also to their parent cell. The first stage in mitotic stage is the Prophase. During Prophase, chromatin condensation occurs. The chromatids will be condensed to form chromosomes. Following that, there is dispersal of the nucleolus. The centrioles duplicates and migrates to the poles. The nuclear envelope fragments and the fragments will be absorbed by the endoplasmic reticulum. Spindle fiber formation occurs during prophase. Following prophase is metaphase. Metaphase is the longest phase in mitosis. Here, the chromosomes are arranged in an equatorial plate or the metaphasic plate. The spindle apparatus is completely organized. In animals, the spindles are attached to the centrioles. The centrioles also have asters, which are fibers which are radiating out from the centrioles. Such mitosis with the presence of centrioles, with the presence of asters, is called as astral mitosis. In plants, Since centrioles are absent, these spindle fibers are attached to the vesicles. Such mitosis is called as Astral mitosis. The spindle fibers are attached to the centromeres and the arms are free. There are two types of spindle fibers. They are continuous spindle fibers and discontinuous spindle fibers. The continuous spindle fibers extend from one pole to the other pole, whereas the discontinuous spindle fibers extend from one pole to the centromere. Here you can see during metaphase, the chromosomes are arranged on the equatorial plate. Following metaphase is the anaphase. That is the shortest stage in mitosis. During anaphase, the centromere divides and the two chromatids, which are now free, they will be moving towards the opposite poles. The chromosomes are pulled to the poles because of the action of the spindle fibers. They move by the action of the continuous and the discontinuous spindle fibers. During this step, the cell elongates as the microtubules lengthen. You can see that the two chromatids have moved to the opposite poles, and these are the spindle fibers that helps in pushing these chromatids to both the opposite poles. Following anaphase is telophase, that is the last stage in mitosis. Here you can see two sets of daughter chromosomes which are found on the opposite poles. Here you can see the chromosome starts to uncoil and become less condensed. The nucleoli will be condensing and reappearing. The spindle fibers which were helping in pulling of the chromosomes during anaphase is no longer required, so the spindle fiber starts to disappear. The nuclear envelope is reassembled from the elements of the endoplasmic reticulum. You can see the two daughter nuclei on both the poles. The third stage in cell cycle is cytokinesis. That is the last stage in cell cycle. In animal cells, cytokinesis occurs with the formation of a cleavage furrow. The cleavage furrow starts to extend from the periphery and extends to the center. And as a result, the cytoplasm is divided into two daughter cells. Whereas in a plant cell, the cytokinesis occurs due to the formation of a cell plate. The cell plate originates from the center and extends to the periphery. The cell plate is made up of Golgian vesicles. The cell plate later forms the middle lamella. These were the stages of cell cycle. To sum up, the cell cycle is comprised of 5 stages. That is the interphase, which comprises G1, S and G2. Mitotic phase, which is divided into Prophase, Metaphase, Anaphase and Telophase. And the last stage is cytokinesis. Thank you.